Welcome to the PubMed Daily Review for April 29th, 2026. Today we scan 9,592 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Generative Artificial Intelligence Transitions Pharmaceutical Development from Empirical Screening to Predictive Molecular Design and Clinical Trial Optimization by Gaith K. Mansour and Hatuf H. Sukkari from College of Pharmacy, Al Faisal University, Riyadh 11533, Saudi Arabia. Let's break down its key contributions. The journey of a new drug is incredibly long and costly, often taking over a decade and billions of dollars, with a high chance of failure. This inefficiency is partly due to the sheer vastness of possible molecules. But now artificial intelligence, specifically generative AI, is changing everything. It's moving us from a trial-and-error approach to a predictive, data-driven one, promising to revolutionize how we discover and develop medicines. This review explores how generative AI is applied across the entire drug development process. It looks at how AI analyzes complex biological data to find new targets, designs entirely new molecules using advanced models like diffusion and GNs, and even optimizes clinical trials using tools like large language models. The core idea is that AI can learn the underlying rules of chemistry and biology to create better drugs faster, forming a continuous loop of improvement. We're already seeing real-world impact. For example, a drug called Renasertib, designed using AI, showed a significant improvement in lung function in patients with idiopathic pulmonary fibrosis. This trial demonstrated a plus 98.4 milliliters increase in FVC compared to placebo, which was statistically significant. The development timeline was also remarkably fast, with a preclinical candidate identified in about 18 months for an estimated cost of $150,000, showcasing AI's potential to accelerate and improve drug discovery. So what's the big takeaway? Generative AI is fundamentally changing drug development, making it more predictive and efficient. However, we must be mindful of potential pitfalls like biases in the data and the possibility of AI generating incorrect information. To truly harness its power, we need to focus on making AI understandable and ensuring high-quality data standards, paving the way for more responsible and effective precision medicine. Up next, our second paper out of 5, Liver Xenotransplantation, from early primate trials to the first-in-human porcine bridging therapies by Alexander Grigory Nastase and Christian Dumitru Lepascu from Faculty of Medicine, Grigor T. Popa University of Medicine and Pharmacy, 700115 Iași, Romania, IGN, NFI, RC, IB, ID, YLN, OGB, CLU, CDL. Let's break down its key contributions. Liver transplantation is a life-saving procedure, but the number of available donor organs is far too low to meet the demand. This shortage results in many patients dying while waiting for a transplant. Xenotransplantation, the use of organs from animals, has long been explored as a way to overcome this critical gap. Recent breakthroughs in genetic engineering, particularly with pigs, have brought this possibility closer to reality. This paper is a narrative review, tracing the journey of liver xenotransplantation from its earliest attempts using primate organs to the most recent groundbreaking studies involving pigs. The authors conducted a comprehensive literature search to synthesize historical data and analyze the latest clinical trials. They examined the genetic modifications in donor pigs and the outcomes of the first human trials, providing a detailed overview of the field's progress and challenges. Early attempts at liver xenotransplantation using primates showed very limited success, with survival often measured in days. More recent trials using baboons extended this to 70 days. Excitingly, the latest pig-to-human studies have achieved a survival of 171 days, demonstrating significant progress. This establishes a proof of concept for using pig livers as a temporary bridge. However, the primary remaining challenge identified is xenograft thrombotic microangiopathy, which needs to be overcome for wider application. The key takeaway is that pig liver xenotransplantation is no longer just a theoretical concept. It has demonstrated potential as a life-saving bridging therapy. While significant progress has been made, overcoming the challenge of thrombotic microangiopathy is crucial. Moving forward, the field needs to focus on making these procedures reliable, scalable, and compliant with regulatory standards to truly impact patient care. Up next, our third paper out of 5, Cell-type resolved acetylation regulator Atlas defines immune endotypes and druggable vulnerabilities in psoriasis by Mengji Xie and Mi Zhou from Department of Dermatology, Yang Hospital of Integrated Traditional Chinese and Western Medicine, Shanghai University of Traditional Chinese Medicine, Shanghai 200437, China, MX, XM, LK, YL, XD, SH, GD, YS. Let's break down its key contributions. Psoriasis is a chronic inflammatory skin disease that often relapses, hinting at underlying epigenetic programs. Lysine acetylation, a reversible modification, plays a crucial role in regulating gene expression and immune cell activity. However, we lack a detailed understanding of how specific acetylation regulators function within different immune cell types in psoriasis. This study aims to map this landscape to uncover new therapeutic vulnerabilities. To address this, the researchers integrated large-scale transcriptomic data with single-cell RNA sequencing. They developed a diagnostic signature based on acetylation regulators and used unsupervised clustering to identify distinct endotypes. Key findings were further validated in a mouse model of psoriasis, and computational methods were used to pinpoint downstream targets and potential drug candidates. The study identified a 13-gene acetylation signature that effectively discriminated between psoriasis and healthy skin, achieving an AUC of 0.886. 16 acetylation regulators were found to be differentially expressed, with HDAC1 showing the strongest upregulation and KD2A the strongest downregulation. Importantly, this signature also separated psoriatic lesions into two distinct endotypes, each characterized by different underlying inflammatory and metabolic pathways. This research provides a comprehensive, cell-type resolved atlas of acetylation regulators in psoriasis. It defines distinct acetylation endotypes, offering new insights into disease heterogeneity and persistence. Crucially, this work identifies potential druggable targets, paving the way for more personalized and effective epigenetic therapies for psoriasis patients. Up next, our fourth paper out of 5, Loss of Immunometabolic Adaptability in MASH, Gut-Derived Signals Drive Macrophage Reprogramming and Fibrosis by Yen Li and Jinxi Wang from Neurology, 3rd Hospital of Shaanxi Medical University, Shaanxi Bethune Hospital, Shaanxi Academy of Medical Sciences, Tongji Shaanxi Hospital, Taiyuan, China. Let's break down its key contributions. Metabolic dysfunction, associated steatohepatitis, or MASH, is a serious liver condition that's becoming more common worldwide. While we have some treatments, they aren't very effective, suggesting we're missing key pieces of the puzzle. Recent research points to the gut-liver axis, and specifically how signals from our gut microbes might be reprogramming our liver's immune cells, driving persistent inflammation and scarring. This paper takes a deep dive into the existing research, focusing on how the gut and liver interact in MASH. It integrates findings about what our gut microbes are producing, how liver immune cells are using energy, and the signaling pathways involved. The core idea is that in MASH, our immune cells lose their ability to adapt metabolically, largely driven by changes in signals coming from the gut. The researchers found that in MASH, the gut microbiome changes, leading to less beneficial short-chain fatty acids and more inflammatory signals like succinate. This metabolic shift reprograms liver immune cells, pushing them towards a pro-inflammatory state that relies more on glycolysis and loses its ability to adapt. This reprogramming was linked to altered key signaling pathways and ultimately drives the persistent inflammation and fibrosis seen in MASH. So what does this mean for us? It highlights that MASH isn't just a liver problem, but a systemic issue involving metabolic dysregulation along the gut-liver axis. This explains why current treatments often fall short. The key takeaway is that we need to think about therapies that target not just inflammation, but also the underlying metabolic reprogramming and the gut microbiome to truly make a difference in treating MASH. Up next, our 5th paper out of 5, Therapeutic Targeting of BCL2 during CAR cell production augments potency through non-apoptotic adaptive changes by Nada S. Aboalela and James L. LaBelle from Hematology/Oncology, Department of Pediatrics, University of Chicago, Chicago, Illinois, USA. Let's break down its key contributions. We know that BCL2 proteins are crucial for preventing cell death, and drugs targeting them are used in cancer therapy. But BCL2 also plays a role in T cells, which are key to fighting cancer. We wanted to see if we could use a BCL2 inhibitor, venetoclax, to make CAR T cells, a type of immune therapy, even better at fighting tumors. To investigate this, we took T cells from healthy donors and patients, engineered them to become CAR T cells, and then expanded them in the lab. During this expansion, we treated some of the cells with venetoclax, a BCL2 inhibitor, while others received a placebo. We then tested how well these modified CAR T cells could fight cancer cells in lab experiments. Our results were very promising. We found that CAR T cells treated with venetoclax were significantly better at killing cancer cells in a dose-dependent manner. They also produced more key immune signaling molecules like IFN and TNF-alpha. Furthermore, these venetoclax-treated cells showed increased resistance to cell death and a less exhausted profile, indicating they were more potent and persistent. This study demonstrates a novel strategy to improve CAR T cell therapy by using venetoclax during their production. By reprogramming CAR T cells, we can potentially make them more effective against cancer. This opens up exciting possibilities for enhancing existing immunotherapies and improving patient outcomes, and warrants further investigation in clinical settings. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.